Köszönöm a kedves meghívást. Szegeden születtem, és 4 éves koromban az USA-ba költöztem. Beszélek magyarul, de kevés a tudományos nyelvtudásom. Elnézést kérem, mert angolul fogok beszélni. So today I want to talk about some experiments we did on the International Space Station. These are collaborations between NASA and ESA. And the topic that I'm studying is tropisms. So tropisms are a directed growth response to an external environmental stimulus. And this panel on the right shows a plant in the environment. So plants have a constant bombardment of environmental stimuli. Two of the most important ones for plant development are light and gravity. And this has been the focus of my research for quite a bit of time. This particularly interaction between light and gravity, or phototropism and gravitropism. As you can see, this topic is not really a new topic. It's been studied for many years. In fact, Darwin and Charles Darwin and his son Francis wrote a book called The Power of Movement in Plants. It's an excellent book, one of his lesser known works. And he talked about tropisms as a modified circummutation. But he pretty much described the need for phototropism, the importance. It is no doubt a great advantage to the plant for their struggle for life to expose their cotyledons to light as quickly and fully as possible. So when a seed germinates, there's gravity that the stem will grow up, the root will grow down, but then phototropism will take over very quickly. Just a quick review, how do plants perceive light? I'm leaving out chlorophyll, but in terms of for photosynthesis, but the three main other categories of photoreceptors are the blue light phototropins, the cryptochromes, and the phytochromes, which are absorbing the red light. We focused on the phytochromes. Phytochromes are a very interesting group because they actually are involved in plant development from seed germination, vegetative development, flowering, and senescence. So one of the interesting things, and the basis of our experiments on the International Space Station microgravity is kind of explained in this slide. So we have phototropism and gravitropism, and it's the interaction that determines the plant growth. So one of the interesting things about phototropism is I can make an extreme statement. You really can't study a pure phototropic effect on Earth because gravity is always interacting with the light. You could study gravitropism, you could turn off the light to study the gravity, but you can't turn off the gravity to study the light effect. And that's why we're doing our experiments in microgravity. We used a laboratory facility that was built by the European Space Agency, and the experiments were done in the Columbus module of the International Space Station. And the device, the incubator we used was the EMCS, European Modular Cultivation System. And this system was really designed for plants, to grow plants, and particularly small plants. And it has a watering system, lighting system. The most important thing, though, is that the EMCS is on, the whole device is on a centrifuge pallet. And this is very important because you could do the experiments in microgravity with the experiments on. But you also could do a 1G control, and you also could do partial fractional gravity, such as the Moon-Mars level. Here's an outline of the kind of experiments we did. A really important point here is that we end up with two types of data from these experiments. We get video downlinks, which are real-time or near real-time from the ISS. And at the end of the experiment, the seedlings are frozen and they're brought back to Earth for biochemical molecular analysis. What this timeline shows, if we start on the left, first of all, we launch dry seeds. And this is really a great thing because seeds are inert. They're living, but they're inert. So you don't have to worry about the effects of the launch in the shuttle era. It's SpaceX, it's about maybe 3 Gs and the vibration because the seeds are inert. The experiments are basically started by watering the seeds, and we have a growth period. We had the stimulation phase where we studied red light, blue light, red light followed by blue light. And we could do a series of intermediate gravities. We could do micro G to 1 G. And this will be important, and I'll explain in a moment. So these are the images of seedlings at the end of the experiment, right before we froze them. So on the left, you can see the seedlings at the beginning of the stimulation phase. We have white light coming from the top. The stimulation lights, the red and the blue lights, in this case it's red, is coming from the left. A few things about space experiments. They're really great fun, very exciting, but they're very complex in that a lot of people are involved. We have biologists and engineers from NASA and also from the European Space Agency. So my role as principal investigator was a lot of times the organizational management. It's even more complicated. The research center we used, where we assembled these experimental cassettes with the seeds, was in California at Ames Research Center. We also had to have a lab here in Florida at Kennedy Space Center. So that introduced some complexity. When I first started this business, we launched on the space shuttle, no longer on the space shuttle, so now we've launched our experiments on SpaceX, which has been a very successful private company. They launch satellites, they launch supply missions to the ISS, and now they also launch astronauts. So the Dragon, we launched the Dragon capsule, docks with the ISS. It stays usually for about a month or so. And then again, the experiments were done in the Columbus module. One concept I'd like to introduce right now, which has been the focus of our experiments, is understanding plant behavior in reduced or fractional gravity. We actually know a lot about plants in microgravity. Plants have been part of the earliest American experiments with Apollo and before. The earliest experiments of the Soviet Union, they launched seeds, and we've had lots of experiments in space. What we know relatively little about, and this is what I'll tell you a little bit more about in this research story, is we don't know that the biology, the behavior of plants in the reduced gravity environments. And as we're exploring the Moon right now and Mars, this becomes very compelling. What are the effects of fractional or partial gravity on planet development? So, of course, Earth, we'll use an Earth-centric definition. By definition, we'll call 1G. The Moon is a smaller body, so the gravitational acceleration is 1/6G, and it is a little bit more on Mars, 3/8G, because it's a larger body. Let me go back to an exciting discovery we made in microgravity, and it's this, the plants exhibited a positive curvature to red light and microgravity. We don't observe this on Earth. Red light has an effect on plants on Earth, but not a directional response. The response for phototropic curvature on Earth is primarily due to the phototropins, the blue light. So blue light is what causes the plant to grow toward the blue component of white light. But nevertheless, in microgravity, we saw this novel response of positive phototropic curvature response to red light. I don't know if people, it looks like people, we had digital stills and we put them together, and you can see how both the roots and the stems are bending toward red light. The other thing you kind of see is something that Darwin talked about is this circummutation. And circummutation is pretty much independent of gravity. It occurs at 1G in microgravity. It's an intrinsic endogenous property of plants. And the tropism is built on top of the circummutation. So, let me explain. I just have a couple of graphs, and I'll try to explain this. So, this is the phototropic curvature in response to red light. The top panel is stems, and the bottom panel are roots. Let me start with the root data, the bottom panel. So what we did was we did the experiment at micro G and 1G, and we saw that in roots, essentially it's a little bit noisy, but there's really no response at 1G. But there is a response at micro G that's very clear. And basically, the Moon, Mars levels are pretty much like 1G in the root. For the stem-like hypocotyls, the story is a little bit different. So there, we have the red light response at microgravity. We don't have it at 1G or the 0.3G. And the 0.1G, which is close to the Moon level, is actually like the microgravity. And these results are very interesting. The other when we had these red light results, people were very skeptical. But we've also shown that mutants, plants that lack phytochromes, those red light receptors, this has an effect on the red light response. So we, in our space experiments, we flew phytochrome mutants. So what you can see in the upper panel, the stem-like hypocotyls. Actually, let me again start with the root. That's the bottom panel. It's a little bit easier to understand. So the wild type, we had a robust red light response. It's pretty much demolished in the phytochrome B mutant. And the phytochrome A mutant actually is attenuated, reduced, but it's not at wild type level. The story in the stems is a little bit different. We have the wild type response, and the Phi A mutant is pretty much like the wild type, but the Phi B mutant actually has this over response. So it appears that the phytochrome in the native wild type plant, the phytochrome B, is actually attenuating the response. But nevertheless, I think when we showed this data, people started to believe us because we saw a red light response, and there are significant phytochrome effects. Let me try to summarize this in a very, very simple way. So one of the things, if we look at microgravity, particularly for the stems, the plants behave kind of like they did on the Moon. If we look at the Mars level of gravity, it kind of behaved as 1G. Some of this was a little bit surprising that the Mars level of gravity was essentially enough to make it Earth-like. And what this tells you is, now this is just for gravity, so if we want to grow plants on the Moon and have greenhouses, there are going to be some issues in terms of lack of sufficient gravity. However, on Mars, these issues seem to largely go away. Again, these are for the parameters we studied. A few other people, there's a German group that studied something like this years ago and had pretty similar results. Of course, growing plants on Mars has lots of challenges, mainly the high radiation environment. But the gravity should not be a problem. Very interesting. So the hypothesis we had about this red light phototropism, how it's masked in 1G, but we could see it in microgravity. If we look at the older plant lineages, formerly called, maybe more primitive land plants such as the mosses and the ferns, they actually use red light in a directional way. It's the seed plants that don't. We don't have much time, and the time is late there, but let me just talk to you about gene expression studies we did, some of the key findings. So first of all, again, we did these kinetic studies of phototropism, but we also, at the end, we froze plants and brought them back. So this is the Melfi, the -80 freezer on the International Space Station. One of the astronauts using it. One of the things that amazes me is, for those of you who have used -80 freezers, you know that they tend to go down and the temperature is raised and you lose samples. But this has been on the ISS for, I don't know, like 15 years, and Melfi has never malfunctioned in a significant way. So we looked at gene expression in microgravity versus 1G for plants. So there are 296 genes were differentially expressed in microgravity versus the 1G control. All about this, but these gene expression studies are very frustrating because you get a long list of genes. What does it mean? So we did this thing called pathway analysis. And rather than looking at single genes, we looked at pathways. And we discovered that there are 8 molecular pathways that were significantly reduced by the microgravity conditions. The one I found, the ones that I found particularly interesting are light-associated pathways. So photosynthesis intending proteins, photosynthesis proteins, porphyrin, chlorophyll metabolism are significantly downregulated in microgravity. And, all the things that I'm doing is pretty much basic research, but NASA is very interested in this because they want to understand how plants are going to be grown in microgravity or reduced gravity, or long-term space missions or colonies on Moon and Mars. This is just an example of the pathway analysis showing how parts of the light harvesting complex are significantly downregulated in microgravity. One of the thoughts I want to leave you with is the ISS as a test bed for Moon and Mars. Even though it will be phased out, there are a number of facilities with centrifuges. Now, there aren't big centrifuges, but there's some small ones. So we really need to use these centrifuge facilities to study development in living organisms at the fractional or reduced gravity. Just another thing I've been working on lately is NASA has this, it's called CLPS, Commercial Lunar Payload Services, where there are robotic landers going to the moon. And we get commercial sponsors. It's kind of a new avenue of funding for me. I've always been funded by NASA or the National Science Foundation by the US federal government. But I've been working with Intuitive Machines, which had a successful landing on the moon, although it did tip over, but it was successful, and we're hoping to develop a small lunar greenhouse. Just a quick summary. So we use microgravity to make these fundamental discoveries about the effects of light on plants to study pure phototropic effects without the complications of gravity. We've discovered some novel mechanisms of phototropism. I didn't talk about the blue light sensing, I focused on the red light. Our studies are among the first to examine plant physiology, plant behavior, plant development in reduced gravity environments. We saw these changes in gene expression, and the pathway analysis is a really good way to go. I think understanding plant biology will be important for Moon, Mars missions. To take a step back, I think a lot of times we talk about building rockets to go to Mars, and certainly we need big rockets. But I think the real questions as we move in the future are going to be in biology. What are the effects of this partial gravity on plants and humans and animals? What are the effects of radiation? I mean, there's serious radiation concerns, and some of the most interesting ideas I've seen for Mars greenhouses is actually the colonies is underground or in caves that protect people and plants from the high radiation. They would pipe in light to the greenhouse with fiber optics. Köszönöm a figyelmet, ha ide engedi kérdéseket angolul válaszolok. I also want to thank some people in my group. One thing I neglected to mention until now is these experiments, the later experiments were done in collaboration with NASA, the European Space Agency, and this is my colleague Javier Medina in Spain, and I was the American investigator who was the European one. We work with people from NASA, European Space Agency. This is just a cool picture. It's one of the astronauts, Tom Marshburn, in front of the EMCS, and the door is open there, and our experimental container is floating in front of Tom. And I want to thank NASA and ESA for funding, and I want to thank you for your attention.
So, my name is Miklós Fárik, coming from the University of Debrecen. So, I'm asking in English, as well as I can continue in Hungarian and many other languages also. Somewhere I picked up the information that you are very proud to be Hungarian origin.
I am very proud to be Hungarian American.
Yes, a couple of months before, we decided that the university, I'm coming from the University of Debrecen, east of Hungary, and we have there space plant growing research investigation under the aegis of the HUN-REN project. So in English I can tell you some information, short information officially about our activities. But before, I also wondering some scientific things about the biological properties of the plants grown on the ISS under microgravity, how the biological modification of bioactive compounds can modify during the microgravity growing of your model plants.
I'm still having trouble. I heard biological modification, ISS. It's still, I don't know if you could hear me well, but
I mean bioactive compounds like amino acid modification of the microgravity or any other modification occurred during the...
Sure. Yeah, I think I understand it, but I think that's a good question. I think there are lots of modifications in microgravity. We focused on gene expression, but when you go downstream to the proteins, there are some colleagues who've studied proteomics and other effects of microgravity. But I guess all I can say is there are multiple effects. If you send me an email, I could also send you some references to some of these other studies. Thank you very much.
Thank you.
Is there anyone from Szeged there?
Van valaki Szegedről, azt kérdezi. Szegedről van
Nincs.
valaki?
Nem jelentkezik senki.
Nincs! Nagyon sajnos.
But there's a lot of people from the whole conference, but now in this room is no. Okay, other questions? Okay. Thank you very much, John. This is a very nice presentation. Köszönöm szépen mindenkinek.
Yes, I'm so sorry. If I would have known about the conference, I would have flown from Florida to Budapest. Although it's very warm now, it's 27 degrees. I think it's colder in Budapest. So, thank you very much.
Köszönjük szépen az előadást.
Igen.
Thank you, bye.